cages for guinea pigs, cameras, and machinery to build his own designs. Lee De Forest, the father of modern vacuum tubes, who was an important contributor to early radio technology, helped to build many of Rife's early frequency instruments. Rife found through arduous experimentation, looking through the microscope as he applied various frequencies, that he was able to kill these microorganisms, proving his theory to be correct. In order to determine which frequencies would kill a particular organism, it was absolutely essential that he be able to see the organism in a living state so that he could observe the effect of various frequencies. Dead microorganisms would show no effect. Rife later called the frequency which killed an organism its MOR, or mortal oscillatory rate. The next step was to solve the problem of high magnification called the Fraunhofer diffraction limit. This theory was that no microorganism smaller than half a wavelength of light could be seen through a microscope. Rife knew from his many years of studying optics at Zeiss that it was possible to build a microscope which could see microscopic organisms as small as a virus. With this goal in mind, he went to work in 1917 and by 1920 produced the world's most powerful microscope. It could magnify with a clarity of 9,000 diameters. Dr. Rife said that this microscope could go clear up to 17,000 diameters, but not with any more clarity. Using this microscope over the next 9 years in his private lab, he advanced further in the field of bacteriology than any scientist had ever done before.
That instrument we used up until 1931. It was a lens microscope, and all of the air was extruded from the body and replaced with glycerin. The lenses were homogeneous with glycerin all the way through the whole thing, and the result was that in allowing the rays to separate and not cross in the interference band of reflection, such as they do in the ordinary standard tube of the regular research microscope. We held them apart, we separated them, and then brought them back together and picked them up again at a needle point. But, as I say, the resolution of this instrument beyond about anywhere from 9 to 10,000 times dropped off decidedly rapidly. We could go up on some occasions, on some preparations, up to 17,000 times, but we didn't consider the resolution anything out of the ordinary beyond about 8 or 9 or 10,000 times, depending entirely upon the specimen we were examining.
Through the 1920s, Dr. Rife isolated many different organisms and was working on tuberculosis. He was successful in killing the rod form with his frequency instrument. But like Bone and Robert Cook before him, who were able to destroy the rod form of tuberculosis with vaccines and antitoxins, Rife's laboratory animals also died. This caused Rife to believe that there was an additional virus that was released when the rod form of the bacteria was killed. In his continual search for a way to see viruses, Dr. Rife theorized that many microorganisms were not able to be seen because they were smaller than the molecules of the existing acid or aniline dye stains. Throughout his work, Rife had noticed that every living thing is made up of chemicals and has its own unique chemical constituency. In his lab, he had been using a spectrographic microscope that could identify the chemicals in an organism by the color refraction index. Knowing that each chemical, when illuminated, gave off a specific color of light, he conceived the idea of staining the virus with light. This dispensed with the need for staining specimens and made it possible to use a frequency of light